Hello, I'm Anna Galetli, and we're going to continue talking about the integumentary system structures. We're focusing in on the last few little parts of the dermis here in part 5. So we're going to focus in on those touch receptors, a little bit about thermoregulation, and then tiny amount on hair and nails. So let's get started on that. All right, so let's focus in on our touch receptors first. I'm going to organize some information, and I'm going to show you one way of doing it. You can do it in other ways. But we're really trying to minimize how much memorization you have to do. So I'm making up a little table. And what I need to know are the 5 basic types of touch receptor and how they are functionally different, okay? If I point out something on a test or I ask what does this root hair plexus or Meissner's corpuscle do and you tell me touch receptor, that's not enough information. I want to know what kind of touch receptor does. What does it do? What is special about it compared to the other stuff? So, first of all, we want to look at location. So, the Merkel cells are found in the epidermis, specifically in that stratum basale, okay? And then I want to compare and contrast the Merkel cell with the Meissner's corpuscle, because what type of receptor is it? Well, both of these are light touch, okay? So how do you distinguish them? So first of all, the Merkel cells in the epidermis and the Meissner's corpuscle is in the dermis, okay? The other thing that's interesting about these is that the Merkel cell, you're finding it in hairy parts of the body. So on the top of your head, in your axilla, on the backs of your arms. Meissner's corpuscles are going to be in hairless areas, like the palms and soles of your hands and feet, the nipples, all right, eyelids, those kinds of places, okay? And that's kind of cool. That's interesting. All right, the root hair plexus. Where is it? Where is it? It wraps around the follicle bulb. So the bulb of your hair follicle in the dermis. Okay? To me, it looks like a gold net in most pictures wrapping around it. Okay? It is a light touch, but it is special because it only detects bending hairs. So when your hair bends, it moves the follicle bulb, and that triggers depolarization with the root hair plexus. Okay? Now, Pacinian corpuscles are very different. In terms of location, they are found in the dermis, and usually pretty deep in the dermis, so getting closer to where the hypodermis is. Sometimes they can actually sneak down into the hypodermis. What's important about them is that they are a different type of touch receptor. They do, let's just actually simplify it. I'm running out of space to write, and here I didn't make my boxes big enough. So we're going to say pressure and vibration. Okay, so very, very different. They cannot do distinct, defined, fine interpretation of touch. They do gross, big, vibrating, deep pressure. Okay, the free nerve endings, you're finding those mostly. Oh, let me change the color. That's awful, isn't it? To go red 2 in a row. Let's change back to blue. You're finding them in the dermal papillae of your dermis. Okay, you also find them deeper down, but that's where we're kind of focusing on them right now. Their job is to detect pain. Okay, when you tear or rip or compress the tissue. It's the free nerve endings that are going to pick up that information and send it back to the spinal cord and brain. Let's look at some pictures. All right, so at this point, y'all have seen this picture a lot. All right, I use this one over and over again because it's one of my favorites. Okay, now I'm zooming this out so that we can see the blue box right here. because there are two things that we cannot see on this drawing. We cannot see the Merkel cells, and we can't see the free nerve endings. It's just not a high enough magnification. What we can see is the Meissner's corpuscle up here in the dermal papilla, and we can see the great big Pacinian corpuscle. For me, Pacinian corpuscles look like tree rings, okay? The Meissner's corpuscle is very difficult to see. You need a just right photomicrograph, and it looks like a faint ladder to me when I look at it, okay? Now finally, and this one's important, I want to use red to point out the strands of the root hair plexus. A lot of people get this wrong when I ask them what it is. So it is detecting the bending of the hairs. It does not move the hair follicle. It is a sensory receptor. It does not move it in any way whatsoever. It detects the bending of the hair. Okay, let's look at a photomicrograph. Okay, so here in this photomicrograph, we can see the epidermis here with the stratified squamous epithelium. Down here is my dermis, and I could go ahead and I can just draw kind of a line. So to me, it kind of looks like something like maybe kind of like this, and that's kind of the boundary between the papillary versus the reticular layers. Okay, so I can see nice thick collagen down here, and I can see it's a lot more faint up here. These folds are my dermal papillae, so I'm just going to do a DP to indicate that. And then if I come right here, I can see this thing that's kind of sticking up in the dermal papilla, and it's going back and forth. Actually, let me do this. It's kind of going back and forth like this, which to me looks like the rings of a ladder, the rungs of a ladder. And that is your little Meissner's corpuscle located in the dermal papilla of the papillary layer of the dermis. Okay, next slide. All right, here we've got a nice picture of the Pacinian corpuscle that is actually in skin. So this is one that's in skin. A lot of the slides we have in our A&P lab are actually in the pancreas. The pancreas has a high number of these, and so they make better photomicrographs. When you're trying to get Pacinian corpuscles, it can be really hard to find the Pacinian corpuscles in skin. So you can see that I had to take this picture from a website rather than from my own lab. And that's just the way it is. So this is my Pacinian corpuscle here. And what you will see is it's got this tree ring structure. So it looks just like tree rings to me, okay? What I really like about this picture is I can see over here my dense irregular CTP all in here. I've got a pretty little artery, but more importantly, I can see two sweat glands right here, and I can also see another sweat gland forming up there. I think that's really cool because you can clearly see that this is skin and not the pancreas. Okay? All right, one or two more photomicrographs. All right, here what you see is the epidermis. Down here is the dermal papilla, and you see this thing that looks like tree roots with little rootlets coming off of it. That is your free nerve ending, which is just kind of cute. And that's the only reason it's up here is because it's kind of cute. Okay, this picture. I need everybody to print this off or use your computer or whatever, but I want you to start labeling it. Okay, and I've got all the labels right up here. Now, because for some reason this keeps importing it upside down, I'm just going to rotate it. And what I want you to do, literally, is I want you to draw arrows or something, and I want you to write what they are. And then underneath it, I want you to say what it does. And I want you to do this several times so that you learn these and you memorize these. Okay? And you can see I've got one right here. Remember, on this picture, you can see the apocrine gland, and then you can see that these are both eccrine glands right here. So remember, on the 3D model, you can tell the difference. On a photomicrograph, you can't. But you've also got that, and you've got that. And then notice these letters. I'm going to erase this arrow so that you can see it. I've got A, and then I got B, C, D, okay? And then down here is E. Over here I have A, right here they're doing A, C, D, E. You need to be able to identify those strata. You've got the papilla, capillary bed, cute little Meissner's corpuscle, etc. Here's another eccrine gland, okay? You should be able to tell that this is the hypodermis. I need you to label this, and I need you to give the functions for absolutely everything. I need you to divide up epidermis, papillary, reticular region, know that this is all of the dermis, etc. ad nauseum, okay? There is a video lecture that goes over this where I talk you through it. You probably want to watch that video lecture. It's a separate one. Okay, let's look at a few more little pictures and talk about thermoregulation. All right, for thermoregulation, we really have, with the skin, thermoregulation in the skin, we have three major mechanisms going on. You have the activity of your eccrine gland, you have the activity of the erector papillae muscle, and then you've got vasodilation with blood vessels. So vasodilation and vasoconstriction. Okay, so looking at this picture right here, we're just focusing in on the eccrine gland right here, which then has a pore that goes up to the skin surface. So watery liquid, when it evaporates, it cools you off, okay? So this one is for cooling, okay? Again, remember this warning. All right, let's look at the next one. All right, the next one is the activity of the erector papillae muscle. When the erector papillae muscle contracts, it causes the hair to stand up on end. In us, on our skin, it looks like goosebumps. If you have a lot of hair, it creates an insulating layer that can help keep you warmer, okay? The secondary effect, all right, so this is the secondary effect, is to cause any of this stuff from the sebum gland to get pushed into the hair follicle and start making its way up to the surface, okay? For thermoregulation, the erector papillae muscles are really awesome on cats, dogs, horses, birds, etc., etc. Anything with lots of hair or feathers. Works a lot less well in humans, except for on our hair. We've got enough hair where we've got enough hair to make an air pocket. But these also, when they contract, create heat. So even if they do not create a good air pocket, they also help to create a little bit more heat, and then your body can also stimulate more muscle contractions, so other muscles, so our skeletal muscles, skeletal, will induce shivering. So erector papillae muscles aren't about shivering, that's skeletal muscles. So remember, this is so messy. Smooth muscle, okay? So that's the second thing involved in thermoregulation. Let's look at the last one. The last one is how we use the circulatory system, how we use our blood vessels within the skin. And this one can be used either to make you warmer or to make you cooler. It can be used for both. So basically right here, you see the blood coming in here, it's coming up here, coming up in here. If it constricts, you're going to be using it to basically divert blood away from your skin to the core of your body, to your heart, lungs, liver, brain, because it's more important to keep those organs warm than it is to keep your fingers and toes warm. So you will divert that blood. On the other hand, if you are exercising and you get really hot, your blood vessels in your skin will dilate, which will take the blood away from your inner core to the surface where it can radiate out like a radiator. And that will let you disperse heat into the environment, helping to cool you off. Okay? So this is a pretty cool little mechanism. All right, I think next we're going to talk really briefly about the hair. All right, super briefly, we got basic functions of the hair. You do not need me to read this to you because you've got it right here. Just remember that some of the functions in other animals, that some of the hair functions in other animals don't really help humans all that much because we don't have a lot of it. So you know that if you're bald, you have to wear a hat because you get sunburns on the top of your head, okay? Dogs don't have that problem because they have enough hair, unless, you know, you're a hairless dog or whatever. A little bit of blunt trauma, it can protect, but again, we don't have a lot, so it's not going to help you the same way it would a polar bear. Little bit of insulation, not super effective. These are probably more important, well, not those, it's these in humans. It's a sensory organ. That's a pretty important thing. We often feel things before they touch us because the movement of air makes our hair move, and we detect that with our root hair plexus. Also the barrier to insects, particles. That's why we have eyelashes. Like, why bother with eyelashes besides the fact that they're pretty? Well, they keep junk from getting into your eyes. Same thing around your woo-woo and all these other places, okay? All right. What's next? Let's just do a really brief picture of the hair. All right, most of this I don't need you to know. I wanted to kind of point out that you have a hair shaft. A lot of us are kind of interested because we talk about cuticles. So the cuticle is the outer edge of the hair. Same thing with the nail. It's going to be the outer edge. And like anything, you've got a cortex and medulla, whatever. One of the things that's really cool is to think about how keratinized the hair is. So skin, oh, that's the wrong thing. So remember, skin is keratinized stratified squamous epithelium. Hair is keratinized stratified squamous epithelium. Nails are keratinized stratified squamous epithelium. So what's the difference? Well, it's the quantity of keratin. So what is the difference essentially between skin, hair, and nails? How much keratin is present? So basically less, most, okay? Then you might be wondering, well, what's the difference between nails and horns? Horns, more keratin, okay? Then you might be wondering, what is the difference between horns and antlers? Well, antlers are bone. Totally different thing. How about tusks? Tusks are actually teeth. Also kind of interesting. So now you know the difference between tusks, antlers, and horns. And then nails and claws. All right, nails and claws are kind of in the same category. Okay, so if we look down here, this picture, we've got the core, you can see it's stratified squamous epithelium. Most of the stuff down here is still cuboidal because it's still alive, and it's getting pushed up towards the surface. I've got the mitotic cells here, I've got melanocytes here, and so forth, and it just pushes it up as it grows. And that's about all you need to know about that. All right, everybody's wondering about hair color, I bet. So here's a summary of hair color. Most hair color is determined by melanin, okay? And specifically eumelanin is going to determine most of it. We do have redheads, which will be have a higher ratio of trichosiderin, and they might also have a higher ratio of phenomelanin. Okay, why do we go gray? Loss of melanin and air bubbles in the hair shaft. Why are you blonde? Less melanin. All right, so basics. Now, do you want to know more about your gingers? Let's go to the next slide. All right, again, I'm not going to read all of this to you because you have it right here and it's fun and you can read through it. I want to highlight eumelanin and phenomelanin. All right, I'd like you to know the difference between those because that's really the difference between brown and black and between red hair color. And what's important about the phenomelanin is to contrast it functionally with the eumelanin. So eumelanin protects you from UV radiation. Phenomelanin, even though it goes and forms an umbrella just like eumelanin, when UV radiation hits it, it releases free radicals. Free radicals punch holes in your DNA. So phenomelanin actually increases potential for cancer. This is hopefully reducing cancerous mutations, which is kind of interesting, okay? So not only do your redheads tend to burn more easily, they are also going to have higher rates of skin cancer, but not because they burn more easily, but because of the whole free radical mess. Okay? Right, one more slide. All right, you can ignore most of this slide. I felt like I needed to say something about nails. So, more keratin. Grows the same way hair and skin do, okay? Down here is your mitotic region. Up here, it's growing out. By the time you get away from your mitotic cells, remember, you can only go so far before you lose oxygen. All of this is dead, okay? The hair itself is also dead, okay? Oh my God. Dead. Hair and nails, dead, except for right by where you're doing mitosis. I don't need you to memorize all of this, all right? It's just kind of neat and interesting to look at. Functionally, I want you to notice the tip of the bone. So the hypothesis is the reason we keep nails, even though we don't really use them for digging or any of that kind of stuff, is it does cause the tip of the finger to be stiffer, and it provides some protection from snapping that bone. That's the hypothesis. All right, that was the end of this part, so you can go ahead and stop and take a break.